Okay, what about the usage of the non-coding DNA in forensic medicine? First of all, why it's important for you? Because it's quite probable that those persons that will get the samples for these forensic studies would be you. So you will get to the patient and ask him to get the sample for DNA testing. Yes. As I'm in forensic medicine, it's of course not only murderers and so on. It's quite rare. It's about establishing parenthood. It's about finding relatives, things like that. A lot of different things. So you will get to the patient and get a sample from such patient, probably. So you need to know what you are doing, and you need to know to answer some basic questions from the patient, because the patient would want to know what is happening. So, what will you do, and what is needed, and how it is achieved? First of all, we need to understand where is the DNA. It's quite basic. You already know that the DNA is in every cell, mainly in the cell nucleus. Not only, we'll get later to this, but mainly in the cell nucleus we have the DNA. Yes, the double-stranded. I'm not good at drawing. So it's double-stranded DNA. Yes. We know also that we have some coding and non-coding DNA. We also know what is coding and what is non-coding DNA. And what we are doing in forensic medicine. In forensic medicine, we need to find the person, whether the person matches some other DNA or not. So, how this matching may look. For example, we have the father and mother, and we are establishing whether a child has similar genes to father and mother. So what we need to find? We need to find similar combinations of the genes, yes, of the nucleotides to be more precise, yes. Of course, getting through all of the nucleotides is impossible. Yes, we have extremely huge amounts of these nucleotides. So we need to focus our search a bit more. We need to focus to such parts of the DNA that is variable. What I mean by variable? I mean, the only information in the DNA is encoded as a sequence of the nitric bases. Yes, so whether this is in such coordination or such different coordination. Yes. So in order to specify that somebody, this is a person A and this is a person B, we need to find some combination of the nucleotides that is different between two people. It would be extremely simple if you would be able to test whole of the DNA strands, yes? But although it's possible, it's extremely hard to do. Yes, the coding whole of the DNA of one person takes really a lot of time, a lot of money. It's generally, from our perspective, point of view, it's possible only for research practices. It's not possible to get it routinely for a lot of patients generally. So, if we can't test whole of the DNA, what we need to do? We need to find some highly variable part of the DNA. What does it mean that it's highly variable? It means that those sequences are changing from person to person. Yes, so some person, all of the people has such part of the DNAs. Yes, these highly variable parts. Yes, and they are called highly variable because we know from the studies that the combinations of the nucleotides in such places tends to be very different from person to person. And why we are doing it just after the topic of the non-coding DNA? Because imagine, if you have some gene, yes, gene that is encoding some protein. You did not yet learn about the mutations, but will manage to do this. So there is some gene that is encoding some protein, yes? If this gene would be very different from person to person, it would lead to very different proteins. And if it would lead to different proteins, in most of the cases, those proteins would lead to sickness. We have extremely well-tuned proteins for the tasks. Most of the changes are harmful. So genes tend to keep very similar composition from person to person. Because if there will be some mutation in the gene, it would quite likely lead to disease or death. So in population, if the people survive, it is quite probable that their genes are highly conservative. So it's very similar from person to person. But in case of the non-coding DNA, if there is some mutation that would hit this part of the DNA, what would happen? Of course, it depends what part of the non-coding DNA. If you would attack the enhancer, silencer, promoter, parts like that, it would have some effect. But in a lot of parts of the DNA, it would do nothing, yes? A lot of the non-coding DNA is just junk, yes? And the organism can afford to have some mistakes there, because it would change completely nothing. And we know where are those parts. So those parts are not genes, yes? They are non-coding DNA. Thanks to this highly variable character, we can get the DNA of one person, DNA of the second person, cut out, thanks to some specialized enzymes, the highly variable parts, and then compare only those highly variable parts. Yes, if those two persons has something in common, they are related. Yes, simple like that. If you would get some non-related people, they would have not much in common here. They would have very similar genes, yes? Because as I said, any mutation in the gene has a high probability of leading to the disease. So genes may be very similar, but those non-coding parts of the DNA would probably be different. Okay, so next question. Of course, naturally, what leads from this topic is to learn about mutations. Yes, so next time explain me what is the mutation, generally speaking. We do not give too much details about this. We will deal with the details and types of the mutations later. But what is the mutation? Yes. So what is the mutation? Give me some definition. How do you understand this? Okay. This is your next task. Next task.